welcome to Who Wants to Be One in a Million?
I'm Bianca Beckley.
I'm Maria Singh, and we'll be your host for today.
Our contestant comes all the way from his dorm in Dixon, Piero.
Okay, so we're gonna jump right in today with the first question, and for our audience, you guys need to pull everything. Okay, question number 1. In the forensic community, what are the microsatellites of DNA that are used for human identification called? A, alleles. B, short tandem repeats. C, complementary DNA. D, RNA.
Interesting. So I did have a cousin in forensics, and she did tell me something. And Mark needs to come down and do the poll everyone. Oh, you have it? All right, we're good. All right, so I did have a cousin in forensics, and she did tell me something about this question, which I can't remember. So I'm going to go on a whim and say it's a B, short tandem repeats.
Whoa, you are correct. Good job, Piero. Okay, so just a little about short tandem repeats that we're going to go over from the module. So your genome contains repetitive sequences of DNA, and these are called short tandem repeats, and they can be up to 1 to 6 base pairs long. And these repeats can occur either 1 time, 6 times, or up to 1 million times. That means there's a huge amount of variation that can occur. So if you're homozygous for a STR, that means that you have the same number of repeats on each chromosome, and then you're heterozygous if it's different on each chromosome. And then we also can use these for human identification because there's such a broad variation in how many repeats you can have. So you can either have 6 or 1 million, and that makes you different from everyone around you. That makes you 1 in a million. And in our lab, we looked at our own STR markers for 10 loci.
Okay, Piero, question 2. What is the ideal length of the primer for the polymerase chain reaction? A, 100 to 200 bases. B, 100,000 to 1 million bases. C, 17 to 35 bases. Or D, 1 to 10 bases.
I didn't see anything there, but to be honest, the 1 to 10 base pair seems really low, and 100,000 to 1 million is, in my mind, way too high for this question. And I don't know why, but C just seems like the right choice because it's the only one not divisible by 10 in all these 4 answers here. And also, one thing I learned from high school is that if you don't know the answer, C for Christ, so I pick C.
Congrats, Piero, you are right.
So a little bit about PCR. It's used to make multiple copies of a specific DNA fragment, and there are 3 steps: denaturing, annealing, and extending. In denaturing, at 95 degrees Celsius, the high temperature, it causes hydrogen bond connections in the 2 strands to break. In annealing, 50 to 60 degrees Celsius, primers attached to a specific location on the DNA template, and in extending at about 72 degrees Celsius, heat is increased so that Taq polymerase binds to each PCR primer and begins adding nucleotides. And so this process is repeated about 25 to 35 times so that each cycle doubles in size and the number of nucleotides, and it could go up to more than 1 million, a lot of millions. A little bit about primers. They're short artificial DNA strands of about 17 to 35 base pairs. I don't know how I got that right. And the GC content, which is the number of G's and C's in the primer, ideally should be about 40 to 60%. And the sequence should be complementary as to prevent primary secondary structures, such as self-dimers and hairpins.
Okay, so moving on to our next question. In gel electrophoresis, which electrode do DNA fragments move towards and why? A, the negative electrode because of positive sugar group on DNA. B, the positive electrode because of the negative phosphate group on DNA. C, the negative electrode because of positive ions in the sugar solution. D, either electrode depending on the fragment size.
Interesting question. I literally only know one thing about DNA, and it's that it's negatively charged. I don't know how I knew that. So, you know, they say negatives, or what am I saying? Opposites attract. So I would say that it's B since it's the only one that's positive electrodes. So I would say B.
Congrats, Piero. That is the right answer. So just to give you a little more about gel electrophoresis, it works really well so you can visualize DNA bands of different base pair lengths. And in our lab, we use a loading dye to keep the samples in the wells and make them visible as they move towards the positive electrode. And the great thing about gel electrophoresis is once you know the distances of the different DNA bands, you can create a standard curve that helps you estimate the number of base pairs in each band, which can help you identify the loci that the unknown DNA bands belong to.
So in our lab, after the DNA extraction, a PCR was run. We examined our PCR results using gel electrophoresis and the chromatogram. And the gel electrophoresis was used in our DNA strand for length, and the chromatogram was used as a visual representation of the DNA. And we used this to calculate our own genetic profile frequency. And so we use a standard curve to estimate the length of the PCR product, and by knowing the base pairs and migration distances and the relationship between them, we were able to find the locus.
And in our lab, we isolated DNA from our cheek cells and then prepared them for the PCR reaction. In order to create as pure as a solution as possible, the DNA was put through 3 buffers: a lysis buffer, a wash buffer, and an elution buffer. And this made the DNA as pure as possible for the PCR reaction.
Okay. So just to talk about like the future implications of our lab. So we learned some important lab techniques such as micropipetting, gel electrophoresis, and DNA isolation. So these can be important in future labs that we work in or fields that you go into, such as research or if you're interested in the medical field as well. And then also there's a lot of innovation today in modern science regarding DNA and DNA isolation, such as gene therapy, genetic modification, and this can be seen in experiments such as knockout mice when you edit certain genes to figure out what's causing a different phenotype. And then we're gonna leave you guys with a fun fact that we share 40 to 50% of our DNA with the cabbage. So today, Piero, you are 1 in a million. Congratulations, you have won.
Thank you.